Hi, everyone. I'm Cassandra Hendrix, a postdoctoral fellow at NYU Langone in the Department of Child and Adolescent Psychiatry. I'll be building off the great talks you've heard so far during this symposium to provide some hot-off-the-press findings that showcase one way I'm interested in using fetal MRI to understand environmental programming of clinical risks. So we know there are general principles that describe neural development on average across people, such as short-term connections in the brain develop first, followed by longer range connections. We also know that each person has distinct connectivity patterns that differentiate them from other people, what's become known as a functional fingerprint. One factor that drives these individual differences in neural connectivity is environmental input. And the first environment we are exposed to is, of course, the womb. Decades of work have tied alterations in the prenatal environment to long-term health and behavior. This began with David Barker in the 90s, who made the observation that infant birth weight was a robust predictor of cardiovascular disease into adulthood. Since then, many independent research groups have repeatedly found that aspects of the prenatal environment, including nutrition, toxin exposure, and stress during pregnancy, are related to long-term child outcomes. My research is largely focused on stress because early life adversity is a quite robust predictor of psychiatric illness. Since the 90s and the birth of these large multi-site consortium studies that probably many people who are watching this are part of, we've seen even more evidence of this relationship between prenatal factors and long-term outcomes. Prenatal stress has been linked to depression across adolescence, as well as ADHD and conduct disorder during childhood. Recent observational evidence also suggests that extremely stressful experiences like trauma that a mother experienced are correlated with psychiatric risk in her offspring, even if those extremely stressful experiences occurred well before pregnancy and as far back as her own childhood. Things like parenting and continued stress into the child's life certainly explain some of these associations. But we also believe this intergenerational transmission of adversity might begin even earlier by programming brain development during gestation. Preclinical and observational studies support this idea that a range of environmental factors, including prenatal stress and childhood adversity, can become biologically embedded to ultimately change the gestational environment in ways that could impact the developing fetal brain. These changes to the developing brain are presumed to be a major mechanism underlying differential susceptibilities to psychiatric illness. But evidence supporting one of the major links in this sequence, that the fetal brain is changed by environmental exposures that are filtered through the mom, is limited. And this is where fetal MRI can offer unique insight into a key mechanism that might link prenatal exposures to long-term psychiatric risk. So to answer this question, I'll be presenting data from the PINK cohort, which is a group of families who live in some of the most disadvantaged, disinvested neighborhoods in the United States. 60% of the sample have a household income of less than $20,000 a year, and they also have a very high trauma load, with over 30% of mothers reporting prior traumatic experiences. These families were gracious and trusting enough to complete a fetal MRI scan during pregnancy, and many continue to be enrolled in this longitudinal study, which is now completing its 9-year visit. So these families are a really unique and important group with whom to study the intergenerational transmission of adversity, and they are precisely the families who are often underrepresented or not represented at all in published developmental neuroscience studies. So the goal of the analyses I'll be presenting today was to examine whether we can see any evidence that maternal trauma exposure from prior to pregnancy is related to fetal brain development. So this is a simplified version of our most recent fetal functional processing pipeline. I won't go through it now, but I do want to mention two things that I think are especially cool. The first is that we've started using a deep learning segmentation algorithm to apply a single manually drawn brain to mask the entire fetal functional sequence, which contains significant motion, as Athena talked about in her talk. We're also very interested in using multi-echo fMRI to improve our denoising, and Amin Majbri from our lab has a poster all about this, so please go check out his poster if this is something you're interested in too. We would love to connect with other people who are interested in multi-echo. So for these analyses, we opted for a functional parcellation strategy that leverages voxel time series similarity to produce homogeneous clusters. Then we used a data-driven approach to organize these parcels into 14 resting state networks, as shown here. The resulting functional networks are consistent with what we would expect to see based on postmortem histological work and prior publications on functional connectivity in fetuses. And what we expect to see is that the fetal brain is predominantly composed of highly modular short-range networks, as you can see based on the similarly colored dots in this image. So because we understand the fetal brain to be highly modular, I wanted to start by examining whether we see mean differences in connectivity strength within networks between fetuses with and without an intergenerational trauma history. And we do see mean differences in the strength of within network connections for 3 networks, even after controlling for fetal age, sex, fMRI quality parameters, socioeconomic status, and prenatal stress. And specifically, we see that fetuses whose mothers experienced trauma show stronger functional coupling within the dorsomedial prefrontal network and within the visual network. However, the influence of trauma on fetal neurocircuitry is not unidirectional, and we see weaker functional coupling within the subcortical limbic network among fetuses whose mothers experienced prior trauma. So rather than uniformly damaging connections in the brain, it seems like this prior history of adversity might lead to a reallocation of developmental processes. It's also interesting that these networks emerged as being related to maternal trauma because after birth, this same circuitry becomes crucial for emotional regulation and threat processing, 2 domains that are disrupted in depression, anxiety, and conduct problems. We can also check whether these mean differences are being driven by a small number of nodes within each network that just have very strong effects. In this connectogram, blue lines represent connections that are weaker among fetuses with maternal trauma history, and red lines represent connections that are stronger among fetuses with maternal trauma history. We can see here that the average within network effects are not being driven by only a small number of nodes within each network. We can also see some preliminary signs that maternal trauma may covary with proto-between network connections, specifically weakening early limbic connections to the visual cortex and strengthening early connection to the dorsomedial prefrontal. So we do see associations between maternal trauma history and fetal resting state networks. These are preliminary findings, though, because our sample size is fairly small compared to the standard in adult imaging. And it's not the case that maternal trauma is universally damaging the fetal brain in these huge ways. The associations we do see are relatively small and they are nuanced. But if you think back a few slides, fetuses were scanned across a fairly wide age range from 25 to 39 weeks of gestation, which basically spans the entire 3rd trimester. In my own work, I've become increasingly interested in timing effects. And I think this is crucial for fetal imaging because the brain is changing so quickly. It's very possible that the gross differences we're seeing in resting state networks are stronger or are only present in fetuses who are a certain age. For instance, maybe the mean differences that we are seeing emerge only later in gestation after neurocircuitry has become more established. Or maybe differences are accentuated during specific neurodevelopmental milestones. Because we're working with a largely cross-sectional sample, it's tricky to estimate developmental trajectories. As a first pass, I was inspired by a clever approach that was used in preterm infants. So the study organized their sample by age at scan, then utilized a sliding window to create overlapping groups of 10 infants to plot neural metrics across the birth transition. I used the same approach in our data, creating 94 overlapping groups of 10 fetuses who were the same age in order to qualitatively examine how within network functional coupling changed across gestation. Consistent with the idea that the brain is undergoing dramatic but uneven neurobiological changes during this period, we see nonlinear changes in network strength across gestation, with each network following its own estimated trajectory. So next, I zoomed in on the 3 networks we found to associate with maternal trauma history to see whether between group differences might be stronger at specific points during gestation. So using the same sliding window approach after splitting our sample by maternal trauma history, it seems as though between group differences are only present at specific times during pregnancy across all of these networks. The biggest difference across these three networks seems to be between 26 and 26 gestational weeks. Interestingly, a prior study from a different research group plotted developmental change in fetal functional networks across pregnancy, and they found that 26 weeks of gestation was this major inflection point for developmental change in functional coupling within the frontal lobe, within the occipital lobe, and for subcortical limbic structures, the three networks that emerged in our analyses with the biggest differences during that same time point of 26 to 27 weeks. So I'm reporting qualitative differences here, but I hope this highlights the idea that our sensitivity to pick up on environmental effects might be heightened if we carefully consider neurobiological timing in our analyses. So we're seeing proof of concept evidence that very powerful environmental exposures are correlated with detectable changes in connectivity patterns in the fetal brain. The changes we see are specific to certain circuits and are nuanced in directionality. I also want to highlight how important it is to consider development on a much smaller time scale than many of us are used to when studying the fetal brain, because dramatic neurobiological changes can happen on a week-by-week basis. And the last point I want to express is how excited I am for others to join this field so that we can work together and produce sample sizes that will be large enough to really dig into whether there are sensitive windows for the transmission of adversity during pregnancy. And we can do the important work of identifying factors that may buffer cross-generation adversity brain associations. And with that, I want to give a huge thank you to Mariah and Tom for organizing the symposium and to all of our collaborators at NYU and Wayne State, as well as the families who make this work